The Firmicutes Bacteroidetes ratio, or FB ratio, can be used as a biomarker for breast cancer. It reflects the balance of intestinal symbiotic microbiota, of which Firmicutes and Bacteroidetes are a major component. The FB ratio has not only been studied in intestinal diseases, such as inflammatory bowel disease, but also in metabolic diseases, such as obesity, non-alcoholic fatty liver disease, inflammatory diseases, and cancers. Breast cancer is thought to be influenced by bacterial extracellular vesicles from symbiotic bacteria in the colon via the gut-breast axis. We investigated this pathway by assessing the FB ratio of the EV microbiome. Because microbial metabolites are loaded into EVs and circulate through bodily fluids, the composition of the FB ratio of EVs is indirect evidence of this pathway. This study was conducted on female patients with breast cancer and healthy controls from 2 hospitals in South Korea. The healthy control group consisted of individuals without any diagnosed disease, and the breast cancer group included patients with stage 0 to 3 breast cancer. Blood samples were collected from participants, and bacterial EVs were extracted from the blood. Next-generation sequencing was performed using bacteria-specific 16S ribosomal DNA. The FB ratio was statistically significantly different between breast cancer patients and healthy controls. The results validate the potential use of the FB ratio to characterize and diagnose breast cancer. The FB ratio in the serum reflects symbiosis or dysbiosis of symbiotic microbiota, which affects breast cancer via EVs. The contribution of each specific bacterium may also have important roles in health. The abundance of Bacteroides showed a close correlation with the fasting glucose levels in patients with breast cancer. Increased fasting glucose levels are a risk factor for breast cancer. The main energy source of Bacteroides is the fermentation of dietary or host-derived glycans. The bacterial compounds formed during this process are commonly found in the human colon and are potentially toxic. They may also have an adverse effect on breast cancer prognosis, and higher levels of Bacteroides may be a risk factor for the development of breast cancer. On the other hand, low-density lipoprotein cholesterol levels decreased with increasing abundance of Firmicutes. Many Firmicutes are butyrate producers that play a role in promoting fatty acid absorption, which may decrease LDL levels. High LDL cholesterol, which is indicative of dyslipidemia, is a risk factor for breast cancer. A relatively low abundance of Firmicutes is likely to have an adverse effect on breast cancer prognosis. The FB ratio can reflect factors such as stage, subtype, family history, and blood test results. Healthy conditions are maintained when the microbiome ratio is balanced. When the balance is disturbed, diseases such as breast cancer can occur. We found that when the FB ratio is 3.37 or lower, the balance of microbial symbiosis is broken, and the risk of breast cancer is increased. Since 50% of metabolites in the human blood are of microbial origin, the change in FB ratio causes dysbiosis in microbiota, inducing an imbalance of metabolites in the blood. This dysbiosis is related to eating habits and lifestyle, which can cause a change in the FB ratio. Therefore, the change in FB ratio could be a trigger for one of the disease pathways leading to breast cancer.